Hello all, welcome back. So in the previous video we've talked about the production of fatty acids and how fatty acids are produced. Now we want to talk about the production of fats. So just to give you a general idea, fatty acids are part of the structure of fats. When we say fats, we mean triglycerides. They are made of two components, the glycerol and the fatty acids. And the fatty acids we've discussed, they are under the influence of the production of myelonyl CoA, which is a precursor discussed in the first video. Now we're going to discuss a very brief introduction about glycerol production and then dive into how the triglycerides are produced from the fusing of both the glycerol and the fatty acid chains. Okay, so here what we will do is basically tell you guys how glycerol is first produced. Glycerol is definitely produced from a molecule called dehydrogenase acetone phosphate. Here, that's number one. That's the first source of glycerol, or it's produced from the breakdown of triglycerides in the adipose tissue. And the breakdown of triglycerides in the adipose tissue, which is the storage place, produces fatty acids and glycerol. So we can use the released glycerol from this reaction into the production of other triglycerides by fusing it with other fatty acids. Okay, so first off, again, for source number one, dehydrogenase acetone phosphate, and lipolysis, which is the breaking down of triglycerides. So now once we have glycerol is converted into an active form because glycerol does not have a phosphate group in its original structure. It should be converted into glycerol 3-phosphate, which is this structure right here, under the effect of glycerol kinase. Okay, so that's the first step. Now we have an active glycerol molecule. Now we have to replace the OH and PO433 minus with fatty acid long chains. And so this is why I wrote here fatty acid 1, 2, and 3. And the structures of the fatty acids that we will be introducing into the structure of the glycerol are listed here. The first one does not have a double bond, the second one does have a double bond, and the third one, no double bond. And so these three are added onto glycerol, and then triglyceride is produced. So the first step here is the fact that fatty acid number 1 would replace the OH here on carbon number 1 under the effect of acyl transferase. So remember, it's called acyl transferase is because we are transferring active fatty acids, which are also called fatty acyl they're called fatty acyl CoAs. Yes, so active fatty acids are called fatty acyl CoAs, and so that's why the enzyme is called Acell Transferase. So we are basically transferring fatty acyl CoAs onto the glycerol molecule, so Acell Transferase. Okay, so here the first step is to transfer fatty acid number 1 onto glycerol, carbon number 1, and so here we get this reaction here. So the fatty acid is added and a CoA is sent out from the structure of the first fatty acid. Remember here, it has a CoA within its structure because it's called a fatty acid CoA. So when it is added onto glycerol, it gives out a CoA, and then the structure of the first product from this reaction under the effect of acyl transferase would give us a MAG. MAG is called monoacyl glycerol, okay, or lipophosphatidic acid. The next product after this, under the same enzyme, Acell Transferase, would also give out a CoA because the second one has a CoA as well, will be added on carbon number 2. So lipophosphatidic acid under the effect of Acell Transferase would produce phosphatidic acid. So the only difference is lipo and phosphatidic. So lipophosphatidic changes into phosphatidic acid, and this has 2 fatty acid chains, and one of them has a double bond. Now for the third step is because we have a phosphate group here, we have to get rid of it first before adding on the third fatty acid chain. So in this case, phosphatase would act on this molecule called the phosphatidic acid to convert it into a tag. A tag is called the triglyceride, which is the molecule that we are actually looking for. So here, phosphate comes out, CoA comes out, a fatty acid is added, and the final product is this one right here. It's called tag triglyceride molecule, or triacyl glycerol. And this molecule will be taken into a VLDL as a triglyceride to be transported back into adipose tissue to be stored. Okay, so this is basically the formation of triglyceride molecule. It has three basic steps. Basically, we're adding the fatty acids onto the glycerol molecule, and everyone has the same enzyme acting upon it to give us this molecule, which will be transported to the adipose tissue for storage by the VLDL, which is a lipoprotein. See you guys in the next video.